Hello, everyone. Welcome to another episode of Science and Technology Q&A for kids and others. This is our 100th episode of this series. I started doing this right after the pandemic hit, and there was a lockdown, and kids couldn't go to school and things like that. And I thought, well, let me provide a bit of a public service and try and do some sort of backup science teaching, so to speak, and try and answer questions about science and technology for kids and others. People seem to like this, and I found it really interesting. And I think I've gotten to be a better explainer as a result of doing this. So I've kept doing it, and hope people are having fun with it. And I'd love to get feedback and input from people. You can find also the recorded episodes of this are available in a variety of formats. Don't ask me exactly what. And there's also a nice website that includes the word clouds from the transcripts of all of these episodes, and you can pretty easily find from those word clouds what kinds of topics were discussed each time. I'm sort of trying to make an attempt to not sort of rediscuss the same things multiple times, because I think we're building up kind of an interesting library of podcasts, basically about well, answering questions about science and technology. All right. Well, with that said, let me jump into some of the questions from today. All right, we have a question from Zayden. How is computation in nature different from computation that a computer does? Well, gosh, that's something I've spent a decent part of my life thinking about, kind of what computation is and its kind of instantiation in different places. So, well, what is computation? Computation is about following definite rules. It's about how you have a structured thing that is, through time, follows definite rules. I think that's sort of the core of the concept of computation, is that you have something defined in some structured way, and over the course of time, it is changing according to certain definite rules. And the thing, in fact, what we've realized from our understanding now in the last couple of years of sort of the really computational foundations of physics, that time itself is really, in the physical world, this process of the computation of the next state of the universe from the previous one. So there's a deep connection between sort of the progress of time and a computation taking place. But when we think about computation as this kind of following of rules with a definite structure, the question is, where does that play out? Well, it plays out in a completely abstract way. I mean, we could just abstractly write down on a piece of paper something where we're taking some particular setup. It might be a line of black and white cells, and we're applying rules at every step that say, if the black and white cells have this configuration, change them in this way, and so on. We could do that on a piece of paper with an ink pen. We could do that in a piece of electronics. We can do that in lots of different places. It's an abstract kind of thing that is just this abstract process of computation. It's just like we can say, 2 + 2 = 4. We can say there's an abstract thing. We can also say, 2 apples plus 2 apples is 4 apples, and we have the physical apples there to count them out. But there's an abstract version of mathematics that is abstracted from the particular appleness of the apples, so to speak. It's just about abstract numbers. And similarly with computation, there's kind of an abstract way of thinking about computation that doesn't have any particulars of what the thing that we're describing is. We're just talking about it as something abstract. So, okay, so what does that mean in terms of how we implement computation? Well, we have sort of a core way of implementing computation, which is using digital electronics, where the things we're dealing with are collections of electrons that represent, let's say, binary bits that build up to all of the data that we store in a computer. We might have billions of these binary bits that make images or make text or make sounds or whatever else. And when I say make, they are the raw material which we then can interpret as images or sounds or whatever. And we have particular devices, whether they're displays or loudspeakers or whatever, that take those raw abstract bits and convert them into something like, pressure waves in the air for sound, or configurations of light for displays and so on. So the sort of instance that we have developed most in technology is having this abstract idea of computation sort of realized in terms of collections of electrons in semiconductors, in microprocessors, with sort of powered by electricity. That's the instance of computation that we are most familiar with. But in fact, this idea of following rules and systems that sort of progress through time by following rules, that's something that is happening all over the place. It's the core thing that's happening in nature. It's the core thing that's happening in the physical world. There are many different possible ways in which you can, in principle, implement that. You can see that kind of application of rules play out. So people have experimented over the years with different kinds of ways to do computation. For example, you could do it, instead of using electrons, you could do it using photons, using light. You could do it using fluidics. You could do it using little fluid in tubes and so on. You could do it using all sorts of different different kind of physical media. You could do it using some mechanical device where the question of whether there's a bit there depends on whether some lever is up or down. There's a lot of different kind of physical realizations of the same kind of computational idea. Now, another place where one can do it is with molecules, where the kind of bits, the data are encoded in whether certain molecules are attached to each other or not, or how the molecules are arranged and so on. And biology and us are really the only great example that exists so far of molecular scale computing, of computing where this abstract idea of following these rules is played out in terms of molecules. That's sort of what a lot of the core of what biology is. Now, if we say, okay, so these computations that are happening, one feature of computations that we typically do on computers is we set them up. We define how the computation should be set up to run. I should say that one of the sort of core ideas about computation is the notion of universal computation, the notion that when you have a system that's set up with particular kinds of rules, you can have it so that by just changing how you initially set the system up, you can make that system emulate the behavior of any other system. So that's kind of the essence of how computers, universal computers, work. You can have a computer that has, it's made from one microprocessor manufacturer or another microprocessor manufacturer. It has different intrinsic instructions in the microprocessor, but by the time you build up the layer of software, the actual functionality that software can execute is the same. It's just compiled differently onto those different microprocessors. And a more extreme version of that is if you were implementing something on a fluidic computer, for example, you would take all those things that you want to, add numbers or something like this, that would all eventually get converted down to these little pressure waves and fluids and an opening and closing of valves in the fluidic system. It will be implemented very differently from the way it's implemented on a digital electronic computer, but nevertheless, the abstract description will be the same. And in addition, if it's a universal fluidic computer, the actual operation of the computer can have a particular form, but by just changing how you set up the initial state of the system, you can have it emulate the behavior of any other system. So you could have it emulate the behavior of some particular digital electronic computer or some other fluidic computer or whatever else. But one feature of the computations that we do on our typical computers is we write programs where we kind of imagine that we know what the point of the program is and how it's going to behave. When we look at systems in nature that we can identify as doing computations, we are typically not the ones setting up those computations to be done. The computation is just happening through the actual sort of process of evolution through time of the system in nature. So we have sort of control over the computations that we're doing in digital electronic computers. That's why and how we set them up. Sort of the intermediate case is molecular biology, where there's a certain kind of understandable narrative and intentionality to what's happening in biological systems, but we're not as directly able to sort of specify the program as we do when we use digital electronic computers. So I think the difference is that there's sort of this notion of computation in the wild, and there's the versus computation that has been domesticated, where we're specifying what should happen, what programs should be being run, and so on. The thing that let's see. So where was I? The question of what of sort of computation under our control versus computation happening in the wild. And we can imagine many one of the things that's sort of interesting, and it's kind of a core idea in the sort of area of quantum computing, is what other kinds of physical systems can we take to use as computers different from semiconductors and electronics. And so in quantum computing, there are ideas about photons, there are ideas about superconducting devices, ion traps, all kinds of different things. You can forget even the quantum aspect of quantum computing, and you can just sort of call it physical computing. And then you're asking the question, well, what about these substrates for computing? Now, most of what's done there is thinking about things where the sort of the structure of the computation is the same as it is for a sort of a big computer, so to speak, or a standard digital electronic computer. And I think the thing that is yet a different idea is, let's, instead of when we sort of have this free running computation in a natural system, that's different from one of these computations where we wrote the program ourselves. Now, we can actually go, and this is kind of the idea of this science that I'm now calling ruleology, of just exploring arbitrary sets of rules and seeing what they do. Then you're kind of jumping to somewhere that is out in the sort of wilds of the computational universe, and being able to run sort of arbitrary computations. And the big question then is, how do those computations relate to what we humans want to do? And what happens is, it's very much like kind of mining physical materials. You suddenly discover, oh, there's this amazing kind of computation out there in the computational universe, and now I realize what I can do with it. You know, I might have discovered a fantastic molybdenum mine or something in my backyard, or a samarium mine in my backyard. And if you don't know anything to do with samarium, then it's fine. You have that mine and it's got something in it, but it doesn't seem very significant. But as soon as you realize, oh, samarium, we're going to be able to use that for this kind of magnet or something like this, and I know what the purposes to which I can apply that are, then it becomes very interesting. And the same is true with sort of computations mined ruleologically from the computational universe. Let's see. Gosh. Well, there's a question here, which I think the answer is rather simple, from Aaron, who asks, why do cars get much hotter than outside air temperature, commenting that their car, its internal temperature was 130 degrees Fahrenheit, while outside it was only 100 degrees Fahrenheit. I think the answer is it's just like a greenhouse, that heat is, there are several different ways that heat can be transferred. What is heat? Heat is just the energy of molecules bouncing around in the material you're dealing with, whether it's air or whether it's metal or whatever it is. Heat is the energy of all those molecules bouncing around randomly inside the material. Well, actually, there's heat is, so how do you transfer energy to something? Well, one way you can transfer energy is photons, light from the sun, taking energy that comes from the sun, and when that photon is absorbed, it will increase the energy of whatever absorbs it, whatever atom or molecule absorbs it. And so when you put something out in the sun, it gets hot because there are photons carrying energy from the sun, and they excite the atoms or molecules in the material, which then run around with larger energy, and that's what makes the thing hotter. Now, what happens in sort of a confusing thing, which is that glass, for example, is a good transmitter of visible light. It's transparent. That's what we use it for, to look through. But what happens is light from the sun goes through the glass. It takes its energy as it goes through the glass. It's the photon that is going through the glass is carrying a certain amount of energy. Then the photon lands on, you know, your car seat or whatever else, and it's absorbed by the molecules in your car seat. And then what happens is that causes the energy of those molecules to increase, so they increase their temperature. But then they, whenever there's molecules running around at high speed and being accelerated and colliding with other molecules and so on, that process will produce photons. But it produces photons that are infrared photons. If you make the molecules hot enough that their kinetic energy is high enough, then the thing will start to glow red hot, white hot, blue hot, whatever else. But at lower temperatures, below where things are white hot or whatever, that might be a few thousand degrees centigrade, would be white hot. Below those temperatures, it will sort of glow, producing infrared light, but at a much lower, at an infrared light that you can't see, that is much lower frequency light. And so the photon is coming from the sun, goes through your glass window, hits the car seat, produces higher energy there. But what you might think would happen if the car seat was kind of out in the open air is that you would kind of, no, if you had a mirror, for example, there, you would be, the light would come in, it would bounce off the mirror, and whatever energy was being brought in by the light would go back out, because that energy would be carried by the photon that was reflected from the mirror. But that's not what happens in a typical material. Instead, what's happening is the material is slightly heated up, but there are no sort of photons like the ones that came in that go out again. Instead, there are sort of much degraded photons that correspond to much lower temperatures, much lower energies. And those photons, they have the following thing happen. Those photons don't get back out. Those photons get stuck, because glass is not transparent to photons of that frequency. It's transparent to visible light. It's not transparent to infrared light, to things that are sort of just things that we can't see in the visible light range, but that are nevertheless other photons that carry energy. So the other thing is, the other way that heat could be sort of, things could come to equilibrium. I should explain one important feature of heat. is that normally, if you have a whole bunch of things and pieces of metal, pieces of whatever, they're all connected to each other. And you heat one of them up. In the usual situation, those things will come to equilibrium all at the same temperature. What will happen is that the molecules, atoms, whatever, in one material, they'll be bouncing around with a certain energy, and they will be at the interface from one material to another. Those molecules will be kicking other the molecules on the other side of the interface. That will heat up the next thing, and so on. And eventually things will come to equilibrium all at the same temperature. The very idea that there is a notion of temperature, that there is a way in which you can characterize sort of the energy associated with, I don't know, a bar of metal or something, as just in terms of a temperature, and you don't have to say, oh, this is the energy of the atoms in this part of the bar of metal, this is the energy in this part, oh, there's this very detailed thing that happens, the molecules are all marching in this direction in this part, they're all marching in that direction in that part. No, they all get mixed up. And if you looked at them sort of under a powerful enough microscope, you'd see that they were kind of bouncing around randomly, and they all bounce around randomly with the same energy. That's kind of associated with the zeroth law of thermodynamics, this idea that you sort of come to equilibrium like that. Okay, so in order to come to equilibrium, there has to be a way to transfer energy around the system to sort of, and usually you can do it by conduction, you can do it by molecules in a solid material, for example, actually sort of hitting other molecules in that material and conducting the heat from one place to another. You can do it from radiation, that is photons being produced that will transfer energy from one place to another. You can do it from convection. Convection is something that happens in gases primarily, where you have, let's say you have your bar of metal and you have some air that gets right next to the bar of metal. Well, then conduction will transfer heat from the bar of metal to the air, but then convection, then that piece of air can just move away. So hot air rises, so it will tend to be once it's heated up, it will tend to rise away from the material. And that's a way that also that heat can be taken away from a hot material. Okay, so usually if you have some system where lots of things are connected together and so on, they'll end up all at the same temperature. And so you might think the air outside the car would end up at the same temperature as the air inside. The reason that doesn't happen is that you can no longer operate, well, convection doesn't work because it's a closed car, presumably, unless you leave the windows open. Conduction, well, it works a bit, but the real driver is radiation, where there's heat coming in through the windows with these photons of visible light coming in, and they are then sort of converted to infrared photons. When they're absorbed and retransmitted, they don't have the same frequency as the original light. But then convection doesn't work, and nor does radiation. You can't get that heat out of the car again. And so what's happening is that the radiation from the sun is increasing the temperature inside, and that heat from the inside can't get out and can't equilibrate with the outside. And that's why greenhouses heat up. That's why cars heat up when the sun is shining into them. Let's see. Let's see. Oh boy, there's a question here, a biomedical question from Alex. Why haven't we discovered a cure for baldness? You know, I as somebody who's definitely afflicted, I occasionally I followed the literature on this subject. I mean, basically, there are hair cells which are continuously producing, normally continuously producing kind of hair follicles, which are continuously producing sort of human hair. And if they die, they don't produce hair anymore. So, can you kind of bring those back to life? Can you make perhaps other cells suddenly decide that they're hair cells? Maybe. There's what is it called? WNT, I think. It's a particular gene that is very much associated with the switching on that gene, I think, is what's associated with the production of pieces of hair. And so if you can switch on that gene in another kind of cell, well, then by golly, you can have hair start to get produced. One thing to explain, a sort of more basic biological point to explain, is we all have DNA in all of our cells, the few 100 trillion cells that we have in our bodies. Every cell has a copy of that same DNA that is the sort of program that defines us, those 6 billion base pairs, those 6 billion lumps of atoms, all in that big long strand that makes a piece of DNA and that defines us. And when our cells kind of, the way that our cells kind of work, in the nucleus of each cell is that DNA, and there's this process that causes different parts of the DNA to be read and turned into proteins. Proteins are the main things that we're made of. Proteins are chains of amino acids. And what happens is the program on the DNA is specifying, this is a gene that might be, I don't know, 1,000 base pairs long, 1,000,000 base pairs long. It's this gene that's made from this chain of amino acids. It's this protein that's made from a gene on the genome, and it's like a sequence of thousands or millions of amino acids long. And those proteins, they're what make us up. They're what make our muscles and our blood cells and our skin cells and our hair cells and all those kinds of things. The sort of the material that builds up those cells is mostly protein. And the different kinds of proteins, there are maybe 30,000 kinds of proteins that are common in us humans, maybe 40,000, maybe a bit more. But they're all just these chains of amino acids. And what's happening is that depending on which amino acids are in the chain, for example, the shape of the final protein will be quite different. Some are long and stringy, like the ones in muscles. Some are kind of folded up and make a little cage, like the ones in red blood cells. Some have different characteristics. And what happens is, depending on which amino acids are in the protein, the thing will have a different mechanical characteristics, and it will fold up in a different way to make a different shape. And once it's a different shape, it will have different sort of ways that it fits together with other proteins, different ways that it can fit together with other kinds of things, and different activities. And one activity is to produce keratin for hair and so on, and to be a hair cell. But the question is, all our cells have the exact same DNA program. How do some of those cells end up turning into hair cells versus skin cells versus, I don't know, kidney cells versus whatever else? That pathway of differentiation, we all start from just one cell, which starts dividing, and eventually we end up with hundreds of trillions of cells. And what kind of cell each of those cells ends up being is an important question. What's become clear in the last 20 years or so is that there are lots of kinds of ways in which the DNA gets modified, the environment of the cell gets modified. It's not the actual original sequence on the DNA, it's additional atoms that kind of hang around the DNA that help determine which proteins will actually be made from the DNA that's in a particular kind of cell. So in any given kind of cell, I don't know how many types of proteins, maybe a few thousand proteins are what's made in a particular cell. And which few thousand proteins are made will differ from one type of cell to another. Okay, why is this relevant to baldness? Well, because if you've got skin cells, and the question is, can you coax one of those skin cells to actually decide, oh, actually, I've decided to become a hair cell. I'm going to sprout a nice, a very fine piece of hair. And so the main thing that one thinks about, I think, is, can one coax those cells to become a different kind of cell? See, one of the things that people didn't know for a long time is people assumed that cells that could turn into any kind of cell, so-called pluripotent stem cells, that the only way you would get stem cells that could turn into any kind of cell was to have cells from a fetus, for example, that were really, in the natural course of events, would turn into a whole bunch of different kinds of cells. People assumed that you had to go back to cells that were there in an embryo to be able to have cells that would have this stem cell feature of being able to turn into all kinds of cells. But it was discovered, 20 years ago now, that actually it's possible to go from something like a skin cell and have it revert. It's still got the same DNA that any other cell has, but it's got all kinds of extra little pieces on that DNA and things that sort of tell it, you're a skin cell. But actually you can get rid of those things and you can have that skin cell revert to being a stem cell. And so when people talk about stem cell therapies and so on, in modern times, what they have in mind is to take things that might be just skin cells and go and revert them to go back to stem cells. And then if you're going to make a therapy where you say, well, I'm going to produce some kind of replacement heart tissue, heart muscle cells or something, or replacement cells of another kind, replacement pancreatic beta cells to make insulin or whatever else it is. What you then want to do is to go back to the stem cell and then say, okay, now go forwards and differentiate, not into, you're now a stem cell, so now turn into the type of cell that you want to have it turn into. And sometimes, like I think many kinds of cells are like a dozen steps away from the original stem cell. So you have to differentiate into this kind of cell, then this kind of cell, then this kind of cell, and eventually you get to the cell that you're looking for. And the surprising thing that I didn't think was going to work, actually, that people have been able to do is to actually get cells to go through all those different steps of differentiation to wind up being a cell that is of the kind that you were looking for. So I think that's one of the main approaches to, get some hair cells back. Okay, the original ones died off, but we can get new hair cells that are that come by sort of going back perhaps to the stem cell stage or somehow otherwise kind of redifferentiating the cell to turn into a different kind of cell. So for example, if you want to produce cells that are it's the same type of story in other places. You can either go all the way back to stem cells, or you can have a cell that was really, I don't know, a liver cell or something, and have it sort of redifferentiate itself to become something like a pancreatic beta cell or some such other thing like that. So that's kind of, I think, the idea, and that's kind of what you have to do is now I suppose there are other things you could do. You could say, if you sort of say, okay, hair cell, you thought you were dying off, but actually we're going to keep you alive in some way, or actually we're going to make another hair cell that is being sort of from that cell, sort of breaking off another a copy of that hair cell. Those might be other approaches, but the thing I've just described, I think, is the most plausible sort of direction, and it's kind of a high wire act of complicated sort of manipulation of how cells differentiate. And of course, one of the issues when you're dealing with sort of redifferentiating cells is, tumors are the result, in some sense, of kind of they are sort of yet a different kind of cell that is being produced from your intrinsic types of cells. But they're a type of cell that has the bad feature that, for example, they don't stop growing. They just keep on replicating, and you keep on getting more and more and more of those kinds of cells. And eventually that takes over, and it's bad for the rest of you, so to speak. And so you have to be careful when you start messing around with how to sort of control the differentiation of cells, that you don't wind up by accident making a tumor cell that just keeps replicating without stopping. The Okay, somebody is commenting, Paul is commenting, compared to other great apes, we've lost most of our body hair. So I wonder if baldness is not just our further evolutionary progression of, and that might be true. I mean, it's kind of like maybe those of us whose hair falls out are the more evolved of this particular type of ape. You know, I do think that you know, one looks for sometimes, there's always a trade-off when you think about biology. Did something happen because of the mechanism of the biological system, or did something happen because evolution carefully controlled the biological system to have that happen? You know, do we end up with an appendix in our guts because it just happened that way, because it was that way in earlier times in evolutionary history, and we just didn't get around in sort of biological evolution, didn't get around to erasing that irrelevant thing? Or is it that way because actually we are fitter organisms when we have that, because, you know, something, it affects our microbiome in our gut or something in some way that's positive, at least if we have certain sources of food or whatever else? It's often very hard to tease apart what happens by virtue of mechanism versus what happens by virtue of sort of a control by biological evolution. And sort of after the fact, you can go back and say, well, that happened because, and sometimes it happened because of mechanism, but then somehow the organism realized, oh, you know, the fact is, because I've got my fingers arranged in this way, so I can peel some new kind of fruit in some different way, and then you're eating that kind of fruit, and then that turns out to be a big win, and so on. But it wasn't that, in a sense, the in a sense that was a side effect of perhaps the, you know, evolution controls things by virtue of the fact that you know, you have some number of offspring. Let's say that purple hair is something that's a really good way to I don't know, not get eaten by predators. And so, you know, some particular parents have some, you know, purple-haired offspring and some green-haired offspring. And it is a random thing that they might have, you know, some fraction of some fraction of just by virtue of kind of the random way in which the genomes of the offspring are determined from the genomes of the parents. In the case of organisms like us with sexual reproduction, there are sort of two genomes that go in, and they get mixed together. There are on every chromosome, there are usually about 5 or 6 different regions, and you'll get, you know, one region will be from the mother, one region will be from the father, and there'll be these crossovers where you're kind of where when replication happens, when the original replication happens, the a part comes from the you know, the father's genome, a part comes from the mother's genome. It's this mixture, and that means that mixing up means that if some part of that genome is giving the program for green hair, some part for purple hair, you'll end up with some number of offspring with green hair, some number of offspring with purple hair, let's say. So then the way that sort of evolution works is, okay, if the green-haired offspring all are big losers because the giant birds come and eat those offspring, then those offspring are sort of taken out of the picture. Only the purple offspring survive. And then when those purple offspring have their offspring, they will have the purple hair genes for sure, not the green hair genes. And so there'll then be more purple haired offspring later on. And so that's kind of the process of evolution by natural selection, by the, you know, the bird coming and eating the green haired offspring or whatever. That's how that works. Now, in the case of humans, evolution has become quite strange, probably, because what matters is basically how many kids do you have, and then how many kids will your kids have, and that will determine, at least in the current stage of human evolution, that will determine kind of what traits win out. So, for example, if it turns out that it's better to not be hairy, because then you lose heat more through your head, and that's good if the climate is warmer or some such other thing. And if there's really an effect that you're more likely to survive to the point of having kids, or you're more likely to have kids if you don't have any hair on your head, then biological evolution will lead it to be the case that there are more humans without hair on their heads. Now, in human society has an effect on biological evolution, because it could be the case that, who knows, maybe the bald-headed guys, get the gals more, and so they're more likely to have kids, and the ones with the giant hair are less likely to get the gals, and so they might just not have any kids. And so then that sort of social dynamic will lead to a biological evolution, so to speak, that the genes that correspond to having the big hair and so on will eventually die out. But so human evolution is now somewhat determined by social forces, as well as by sort of what might be seen as more pure biological forces. Now, the same is undoubtedly true for all other organisms. I mean, there are endless organisms that have elaborate mating rituals and mating displays and so on. And although we don't really know quite what the fashion is in peacocks and so on, one can imagine that the same types of things that play out in human social dynamics and sort of its effect on human evolution also plays out in peacock dynamics. Whether and maybe there's a season for maybe there's a few decades when it's really cool to be a super thin peacock, and maybe there are other times when it's really cool to be a really rotund peacock and so on. Maybe that plays out in other organisms as well. But I think in this now, it gets much funkier with human evolution if we start doing gene editing in humans, and if people start saying, gosh, it's really cool to have green eyes, let me just change my genome or change my children's genomes by editing the eye color gene and just change it to be green eyes. And that's something that in the last decade or so, it's been known in principle how to do that, although actual therapies based on that are just starting to be used, some for some eye diseases and so on, where you actually can go in and you're delivering something that actually goes in and edits the genome and changes and then changes the fundamental program that your cells are running. So, but if that becomes something that is a thing that is routinely done with humans, and by the way, by the time you've sometimes you can change that genome for cells that are kind of late in the stage of replicating, where they're not like stem cells or something, but you can also imagine making those changes right at the beginning, and the original cells that replicated to make a human, and then you can expect that those changes will be passed down to the offspring of that human, and so on. And so you've actually made a permanent change to the germline of the actual genes that get passed down. There have been proposals to do this for things like mosquitoes to prevent malaria and so on. It's a little bit of a be careful what you're doing type situation to have to actually make a change in a population, and particularly when you set things up so that you kind of drive the change into the population, so that more and more mosquitoes will end up having these modified genomes. it's like, things could go horribly wrong if you made a mistake there. And it's often hard to tell. This is kind of a fundamental feature that comes from the sort of computational character of biology, that it's hard to tell what a particular change will lead to in the end. You think, oh, I'm changing it for this purpose. But the problem is, just like software and programs and you can have bugs in programs. You thought you were writing a program to do this, but the computer did exactly what you told it to do, except there was something you did not foresee, and that caused it to do something you didn't want, that you would call a bug. So I think, okay, I have various comments here about how to describe the mating of humans. I'm not sure I have much to comment on that. It's always a question to what extent we humans differ from sort of other species in what we do, and is the mating display of the humans really any different from the mating display of the peacocks and so on? But what seems to differentiate our species from others is the kind of, what seems to differentiate us is our ability to pass on things other than genetic, other than genetically. In other words, the fact that we have language that allows us to describe things abstractly, that allows us to take things we learnt and pass them on to the next generation, so to speak, and to do so with this sort of abstract way of passing on thoughts. That's something that doesn't really seem to exist elsewhere in living systems, or at least we don't recognize it. Certainly, there are things where there's a small amount where it's like, okay, you're a bird, you should learn to fly, or okay, you're a whatever, you should learn to hunt. It's kind of like, watch what the parent does and emulate it, and you learn from that. But this concept that we humans have of taking kind of abstracted ideas, sort of pure thoughts, and being able to communicate them to later generations, that seems to be pretty unique to us humans from what we can tell. And sort of the key invention that has made that possible is human language. Because the way to think about it is, in a brain there are thoughts going on, and those are associated with the little electrical impulses in our nerve cells and all that kind of thing. But everybody's brain works slightly differently. And so the actual pattern of electrical impulses, it wouldn't be very useful to take the pattern of electrical impulses in my brain and kind of transfer it wholesale to another brain. If what you're doing is showing a physical motion, what do you need to do to swim? What do you need to do to fly or whatever? Those physical motions, the parent bird and the child bird will flap their wings in the same kind of motion in order to fly. They may have slightly different, feathers in their wings, but basically I don't know, what is it? Monkey see, monkey do, or whatever. You can explicitly see what you're supposed to do because it's a big physical motion. But when it comes to thinking, even if we had some kind of visual display of kind of the patterns of electrical activity in our brains, it wouldn't do the other person much good to see that stuff, because their brain is different. Thinking is this very detailed thing. You couldn't just sort of transplant the pattern of neural activity from one brain to another. So we have to have some sort of more abstracted description of things that is transportable from one brain to another. And that's kind of where I think the notion of language and the idea of sort of robust concepts and things like words come in. Because, we say the word cat, and that corresponds to some particular neural pattern in my brain. It'll be different in everybody else's brain, but yet the notion of cat is something which we can abstractly communicate from one brain to another, even though the details of where the neurons are firing is different. And I think that's sort of the key idea that has led our species to be able to do the things it can do is that we have this kind of layer of abstraction that's captured in language, captured in things like words and so on, that allows us to transfer knowledge from one detailed knowledge from one organism to another, from one generation to the next, and so on. And that's sort of a crucial thing that has happened there. Well, let's see. Oh my, lots of kinds of questions here. Let's see. RBS is asking about things in nature operating according to rules and why different flowers have different shapes and colors and so on. Okay, so biological organisms, one of the big things they do is grow, and different kinds of organisms have different sort of big ideas about how to grow. So for example, plants, they have a cell and they make new cells, but the walls of plant cells are rigid. They have cellulose in them. And that means that if you want to grow as a plant, you have to just sort of add things on. You grow the big trunk up higher up for a tree. You don't sort of just add in more tree cells into the trunk. You're just always growing upwards, so to speak. For us animals, we're more squidgy, and we can grow internal. We can have cells inside us where there are more of those cells, and we kind of, that thing just sort of pushes other cells out of the way. So there's this question of sort of what rules are used for which plants to make the shapes they make, and so on. There are some sort of bigger general rules, like plants tend to use branching and so on to make their structures. But the question of which branching you use, well, that becomes a more detailed question. The plant has sort of a big idea, but then there are more detailed things controlled by the particular DNA of a particular plant. Now, one of the things that's confusing about that is the way that geometry works out. You can have a plant which is branching, and it's making its branches, make one set of branches, they make branches, and so on. Depending on the detailed angles of those branches, the overall pattern that can be made can be very different. If the branches really are sort of at big angles, you'll end up with something which is a very kind of separated little fern-like thing. If the branches are at much smaller angles, things will join up and you'll end up with something which is a much more sort of entire thing, like a leaf or something like that. So while it seems like that to us, it looks like, oh, the plant did something really different, at some level, it's actually very similar. It's just a very detailed change of the angle that something branched out at will lead to a really big change in the actual structure of the grown plant. So I think what tends to happen with across lots of biological organisms is that a class of organisms will have one basic idea, like we're going to do branching. And then different organisms, different species will have detailed differences in how that works. But those detailed differences may visually to us look like they're really different. And moreover, to the actual operation of the plant or animal or whatever it is, it may have a big effect, because while it's a small sort of effect to go from having your skeleton be such that you routinely walk on 4 legs, that you gradually change, the shapes of bones and so on, and eventually, oh, we can walk on 2 legs, and then quite different things become possible. I see a question here from Erin about plant cells, since we're talking about plant cells and Voronoi meshes and animal cells and so on. So, okay. So one question is, what shapes are the cells? So if you look at your skin, you will see, you can see the individual cells on your skin, and they are, they're different shapes. They're often, and skin cells, they're often kind of like a parallelogram type shape and so on. They're different shapes. If you were to look at the cells in the back of your eye, you would see kind of a hexagonal array of cells, I think, and different kinds of things. But these cells, they end up being different shapes. You have a whole bunch of cells, they're all sort of smooshed together. What shape do they make? Well, that's a good question. And there are, if you just imagine a bunch of cells that are just sort of smooshed together, you can imagine taking some clay or something like that, just making sort of clay balls and taking all these clay balls and just smooshing them all together. And what kind of shape do they, and pushing them so they're no longer spheres anymore. They distorted a bit, and you kind of crush them so that they're all fitting together. What shape do they make under those circumstances? Well, actually, people didn't know that until in the 1940s and 1950s. People did experiments, the rather obvious experiment I just said. They smooshed together a bunch of clay things. They took it apart. They answered questions like if you have one of those clay balls, how many other clay balls were kind of right around it, touching it. So a thing to know is that if you have circles and just in a plane and you smoosh the circles together, they'll form always the same pattern, a hexagonal pattern. Around every circle, there'll be exactly 6 other circles. They're all touching it. Those 6 circles are all touching 6 circles. You get this hexagonal pattern. It's kind of like a beehive, etc. That's a typical form that two-dimensional things smooshed together make. And so, for example, when you have in plants, there are places where there are sort of cells that are fibrous cells, and they're smooshed together, and it's kind of like a planar thing like that, and they'll tend to make these hexagonal patterns. When you look at the three-dimensional case where you've got these balls being smushed together, well, let's talk about spheres to begin with. If you take spheres, the closest packing of spheres is the one that people tend to use to stack up fruit in grocery stores, the so-called Kepler packing. And how many is it? I think it's 20. I think every sphere has 20 other spheres that touch it. I think that's right. And that's for a long time, it wasn't known whether that was the closest packing, whether that was the packing of spheres that would minimize the amount of empty space between those spheres. A chap I know named Tom Hales proved in the, what is it now, 15 years ago, 20 years ago now, proved that mathematically that really is the closest packing. That had been sort of an open question since the 1600s when Kepler first started really talking seriously about this. Was it really the closest packing? We know that in 2 dimensions, the hexagonal packing is the closest packing. In 3 dimensions, what is the closest packing? It was a very difficult proof actually done with our Mathematica software system. And then kind of people didn't really believe the proof because it was such a long and complicated proof, hundreds of pages long. And Tom Hales, the person who had done it, eventually said, look, you don't have to believe me. I'm going to make the proof completely computational. I'm going to make it so that every step can be immediately verified by a computer. There are no words involved. It's all computational language. That took him a decade. Sadly, I'm not sure how many people really cared about that. It's like, well, okay, now it's completely nailed. There's no question about it. This proof is right. But people kind of believed it anyway. Although they said, oh, no. You really have to try and, give evidence for it. And what they ask for is not necessarily what they really want. But in any case, so it's known that packing, the Kepler packing, is the closest packing of spheres, of equal size spheres. If you say, well, what happens if the spheres are of unequal size, for example, still not known what the closest packing is. It's not even known for the 2-dimensional case where you have unequal size circles, quarters and dimes or something. What's the closest packing? It isn't known. It's not known even whether that's a regular packing that looks like kind of a crystal where around every coin is the same configuration of coins. It could be sort of a random packing that is the closest packing. It's not known. Now, by the way, if you take spheres and you just sort of throw them in a ball pit and you say, how close can you pack them? They will typically not pack in the closest packing. However hard you smoosh them, you won't manage to get them to the closest packing. In 2 dimensions for circles, that does work, but in 3 dimensions, it doesn't work. You end up with these situations where things are jammed and you can't get there from here, and you have to kind of shake the balls. And that's what happens when you're kind of trying to take some coffee or something and you're trying to push those coffee beans down. You shake it a bit, and that causes the places where the coffee beans, coffee grains are jammed. Causes them to kind of jiggle around, and then they can get closest to the closest possible theoretical packing. Okay, so that's what happens with sort of rigid balls. Is there's a closest packing which you don't achieve except by carefully putting things that way, or sometimes by jiggling it around enough. But what happens with these kind of clay balls or something like this that are still flexible? Well, what happens is that the thing you get are tetrachydecahedra. Tetrachydecahedron is a 14-faced solid. Well, okay, that's a very weird thing, because the sort of the most famous polyhedra are the Platonic solids. It goes tetrahedron, cube, octahedron, dodecahedron, icosahedron. There are only 5 of those Platonic solids. Those are solids where every face is identical. So in a cube, there are 6 identical square faces. On a tetrahedron, there are 4 identical equilateral triangle faces. On a dodecahedron, there are 12 pentagonal faces, and so on. Those are the only solids which you can get if all the faces are exactly the same. If you and they're also convex; they don't have any indentations in them. If you keep going and you say, well, actually, I'm going to allow different faces. I'm going to allow 2 kinds of faces, or I'm going to allow indentations, and so on. There are many more polyhedra. In fact, you can invent a polyhedron where you're just putting a bunch of points down in space. And for example, let's just imagine you just have points in space and you put shrink wrap on top of those points in space. That will so every time there was an interior point, you wouldn't reach that point because the shrink wrap just would not go there. But you shrink wrap sort of the outer boundary. That's what's called the convex hull of the set of points, is that outer boundary that you get by doing shrink wrapping around them. But you could also try to sort of approximate what's the way that you would sort of have webbing on top of those points that you have. That would make you a different kind of polyhedron with those points, for example, as, let's say, corners of the polyhedron. So in any case, there are many polyhedra you can make. But what happens when you smoosh clay together like that is you, on average, get 14-faced polyhedra. Those are not regular polyhedra because there are no regular 14-faced polyhedra. There's the dodecahedron with 12 faces. There's the icosahedron with 20 faces. There's no 14-faced one. So those are irregular polyhedra. Their faces are not all the same. But on average, the faces that you will get these 14-faced things. And I think that there's you will get a distribution of sizes of faces. Let's see, there is a thing, what is it called? The Aboa-Weir law, I think, is one that describes the sort of the distribution of numbers of faces, faces in that particular case. There's a sort of a related thing. If you are doing, okay, let's talk about the analog of shrink wrapping or something, let's say for points in the plane. This gets us to this thing called a Voronoi diagram or a Voronoi mesh. Okay, so imagine you have a bunch of points in the plane. Let's imagine that those points are, I don't know, shopping centers, okay? And you want to ask the question, you've got shopping centers all arranged in different places. And you want to ask the question, for any given person, which shopping center will they go to? If they're closer to one shopping center than another, let's assume that they'll always go to the closest shopping center. And then what you want to do is draw a map that shows around each shopping center, where do the people who will go to that shopping center live? And that will be those people who are closer to that shopping center than to any other shopping center. And so what you can do is you can kind of segment a map by, this is the region for this shopping center. This is the region for this shopping center. All those different regions fit together. they all have, in fact, in the case I just defined, they'll all have straight edges, because if you imagine between two shopping centers, you're asking, if you look, let's say there are only two shopping centers in the world, what will be the, and assume that the Earth is flat, so we don't have to worry about spheres and all that kind of thing. You've got these two shopping centers, and you ask the question, what region do people live in that's closer to one shopping center than the other? Well, it's clear you can just make a line, the bisector of the line between those two shopping centers. It will be this line that just goes sort of equally between the two shopping centers. It's a line that divides the people who go to shopping center number 1 versus shopping center number 2. You generalize that when you put down many shopping centers, and there's this whole network of lines that sort of mark the distinction between one shopping center and another. And by the way, you can look at this in reality. You can look at things like commuters, people living around cities, which city do they commute to? And you find that this kind of arrangement of sort of the segregation of things by sort of this mechanism is quite prevalent in real life. These things are usually called Voronoi diagrams or Voronoi meshes. This arrangement of lines that defines these regions closer to one point than to other points. And people, there are all sorts of strange things in the sort of theory of geography. There's one, it's called what is it called? It's called the law of retail gravitation, I think, which is basically says that people will tend to go to people will tend to go to the closest shopping center. It's a little bit more to it. And for a while, people were really pushing the idea that there was sort of an inverse square law of attraction to things like shopping centers, which I think was a bit confused because they were sort of imagining that physics had a universal result there, which isn't true at all. I mean, that the result in physics has to do specifically with gravity, specifically in 3 dimensions, and so on. So maybe the space shopping centers where things are arranged in 3 dimensions, it might be more relevant, but not so much for the 2-dimensional case. But in any case, so that's the idea of a Voronoi diagram. You can do the same thing in 3 dimensions. You have all these points laid out in 3-dimensional space, and you say, what's the region that's closer to one point than to any of the others? And it will make a polyhedron. It will make an irregular polyhedron. And so that kind of thing is what you would potentially get by sort of smooshing together these balls of clay. I don't think you quite get that, because what happens is, if the balls of clay are of roughly equal volume, that will tend to keep those points far away. So if you put down points at random, then you will get this, and you say what's closer to one point than the other, then you'll get this Voronoi mesh. But if what you have is physical balls of clay, then each ball of clay will tend to be sort of a roughly equal distance away from another ball of clay, because there's just the same amount of clay in each ball. But I think that what you end up getting is, instead of everything having exactly like 14 faces or whatever, there's a distribution of numbers of faces, and that is what gives you, that's sort of characteristic of what you get in that situation. And it turns out to be a pretty good model of the way that cells in biological tissue arrange themselves. Now, there are a bunch of similar kinds of things. In the 2-dimensional case, soap films or soap froths are another kind of thing. If you put a bunch of soap in a sink or something, and you see all the bubbles there, and you'll see these bubbles, and the bubbles, each bubble will have a certain number of, it'll be arranged with a certain, they'll tend to be roughly equal sized bubbles, because of the way surface tension works. And these bubbles will tend to arrange themselves to eventually be roughly a hexagonal array of bubbles. There is a rule that tells you the average number, the distribution of numbers of faces of those bubbles. So you could have them all be perfectly hexagonal. So it's like kind of like a beehive type setup, a honeycomb rather, I should say, type setup of hexagons. Or you could, but sort of the naturally occurring soap froth will have not quite arranged itself in that honeycomb type fashion. And if you start looking at the distribution, the numbers of sides, some of them will have 5 sides, some of them will have 7 sides, 8 sides. There's a definite distribution of numbers of sides. I think John von Neumann was involved in figuring out that that at least observation, in his case from metal grains, grains in a metal that had come from the cooling of a liquid metal. So in any case, actually it's interesting that rule. Okay, so that rule is what comes out, the number of sides that you get in the soap film. If you look at a soap film, what happens is there will be bubbles, there will be two neighboring bubbles, and they'll suddenly, the line between them will suddenly disappear and they'll merge, and then the other bubbles will sort of push on those bubbles and make them closer to the sort of the average size. And it turns out there are particular sort of moves that you can identify. If you look at the borders between bubbles, there are particular things that happen. Like there's one where you have sort of the 2 goes to 1 goes to 2 configuration that turns into a 2 at a V and a wedge with a line between them. One of those transforms to the other. You can think about these like transformations on graphs. If you imagine just a pure network, if you imagine the network of the borders between bubbles, you can draw that network and you can imagine transformations on that network that are associated with kind of the coarsening of that foam formed from all of those soap bubbles and so on. So in any case, the result of sort of randomly applying those coarsening transformations is what gives you that distribution of numbers of sides. One thing that's actually a bit surprising is it seems like a rather elementary mathematical kind of construct of, you're just taking this graph and you're coarsening it and so on. I have never found a simple derivation of the results about this. The only derivation I know goes all the way through conformal field theory and Feynman diagrams and so on, and loops back to talking about networks that you get either abstractly or in soap foams and so on. It's a bit of a surprising thing, that you have to kind of go out to really advanced math and come back again to get that result. But in any case, that same result, that same kind of result applies to the three-dimensional case and sort of smooshing together things like biological cells. Now, another thing that might be mentioned there is that process of kind of smooshing cells together. It's a little bit like what happens in liquids. So, when you have a solid that forms a crystal, all the atoms are arranged in some very regular way, and it's pretty typical to get a hexagonal close-packed crystal, for example, where you have the atoms arranged in a hexagonal array. You can get cubic crystals like table salt and so on. You can get different, there are 26 kinds of crystal structures that you can get where you are arranging these atoms and collections of atoms in this regular array. But solid crystals, the atoms tend to be arranged in these regular arrays. In gases, the atoms are just bouncing around randomly. In liquids, it's more like the balls of clay smooshed together. You've got all these molecules in, let's say, water or something, and they're all kind of smooshed together and colliding all the time. And it's a little bit more like what you have in the case of the balls of clay. And so you can ask the question, well, for example, what's the typical kind of configuration of things? Like if you imagined around every molecule, how many molecules sort of touch it? Imagine a polyhedron that you say, this is my territory as one molecule. Where are the nearest molecules to that territory? Well, one of the weird things that happens in crystals, in liquids rather, is that you quite often get you get kind of icosahedral, sort of effective icosahedral structures. Okay, why is that weird? If you imagine taking cubes, for example, you know that you can just stack up cubic bricks and you can make any sized object from them. If you take, let's say, certain kinds of, you take hexagonal prisms, you can do the same thing. You just sort of put the prisms together in a pavement and then stack those up. That's kind of the structure of graphite, for example, is carbon atoms arranged that way. And there are other shapes. There are a limited number of shapes. There's a, oh my gosh, cube octahedron is another shape, which is an octahedron with its points sort of chopped off. That's another type of solid that fills space. You can just pack them all, identical solids like that, together to fill space. And those are the configurations. It's those things and those kinds of packings that lead to the structure of crystals. But in a liquid, you don't have that kind of regular packing. You have all these things kind of jiggling around and getting smooshed together, a little bit like those clay balls I was talking about. So the question is, what shapes do you get? And the weird observation is that you sometimes get kind of approximate icosahedral shapes. If you take icosahedra, 20-faced polyhedra with triangular faces, you take a bunch of icosahedra, they don't fill space. You can't pack them together. They've got holes between them if you try and pack together regular icosahedra. But if you're just doing this jiggling around and sort of randomly having molecules kind of just be nearby for a little while and then go away, as they do in a liquid, then you can get something which has an approximate icosahedral structure. And that seems to be what happens to some extent, at least in liquids. Different liquids work differently. Water is an incredibly complicated case. Water ends up with these clusters of molecules. It's not very well understood yet. These kind of clusters of molecules that are very organized within the water. It's like miniature little pieces of pseudo solid that exist at a very, very microscopic level in water that progressively get broken up and reformed and so on. But water has a particularly complicated structure as a liquid. But that's kind of the idea, is that there's this kind of not imprecise kind of smooshing together of different parts that lead to certain forms of geometrical structure that are different from what can be achieved if you really want to fill up space and so on. Let's see. There's a question here from Universe. Do you believe that a concrete description of evolution is waiting to be fleshed out within the multicomputational paradigm? And if so, does its basic rule relate to expansion of hypergraphs and so on? So what's being referred to there is our models that have arisen from trying to understand fundamental physics. And let me not go into a big description of that here, but suffice it to say, one of the key ideas is this notion of multicomputation. There's the idea, there's a definite rule you can follow and you go from one situation to the next, to the next, and to the next. But imagine you have a game like tic-tac-toe, for example. At every step, there are many possible things that could be done next. You can make kind of a graph of all those possible histories for tic-tac-toe. Sometimes you'll have at a particular board state, you'll have two different things that can happen. That's a branch in the history. Sometimes you'll have two different board states where you fill the board in and they end up looking the same, even though they came from two different states. That's a merge. You get this multi-way graph that describes sort of the set of all possible histories of how a tic-tac-toe game could be played. That's kind of a multi-computational phenomenon where you have all these different possible paths of computation that are entangled with each other. So a question is, what about biological life? What about evolution? Well, it's pretty common to draw this thing that's usually called the tree of life, that imagines that you start from the very first organism and it gradually evolves into, a fish and a lizard and so on. And you've got this big branching of all these different possible forms of life, starting with the very earliest form of life. Presumably life only arose once on Earth, who knows? At least maybe only one of these origins of life survived. So you end up with this sort of branching tree as different species get produced, as what happens is, I was talking about the green-haired, the purple-haired creatures, maybe what happens is that after a while, the green-haired creatures only mate with green-haired creatures and purple only with purple, and you get these sort of separate species formed of the green ones and the purple ones. So that, and sort of the way you imagine life on Earth kind of emerging is this, you talk about this kind of large-scale tree of life, where you're sort of branching off all these different species that get produced. I'm more interested actually right now in a more extreme genealogical tree of life. I mean, if you draw your personal genealogy, you have two parents, there are four grandparents, there are, grandparents. Eventually, maybe some of those grandparents will be the same people. So it isn't quite a tree even going backwards, so to speak. And you might have some number of children, and they have children. You'll get kind of the tree going forwards. It isn't perfectly a tree because, you know, when you have distant enough cousins, they might be marrying each other, and then you'd get sort of a merge in the tree and things like this. But you can make this big genealogical tree, for example, for humans, and you know, the 100 billion humans who've ever lived, there is a definite genealogical tree that connects all those humans. You know, in Iceland, for example, I think the complete genealogical tree going back to 1000 AD is known, but in most parts of the world it's not known, although one can piece together some approximations to it. But in any case, let's you imagine this completely genealogical tree of all the relations between all these organisms, all these humans in that case. But imagine, let's scale that up to all organisms that have ever lived on the earth. And, you know, there's some that have asexual reproduction. There's some, you know, where there's just one parent producing many offspring. There's ones where there are two parents and so on. Like for you know, sort of all the modern creatures. Then you can imagine this whole sort of, it's actually a graph of relations, genealogical graph of relations in all organisms that have ever lived. I don't know how many there are. I was trying to estimate that recently. There's maybe a trillion, roughly, organisms that have lived on the earth since the origin of life 3 billion years ago or so now. And so you've got all those relations. And one thing that you do is in that graph, you can kind of identify regions of the graph that correspond to different species, regions of the graph where there really isn't much cross-connectivity because there isn't mating across species. That's kind of the typical operational definition of a species, is that it only mates within the species and not to other species. And so that has some, so what's happening is this giant genealogical graph is broken into these sort of blobs that correspond to all the different species. And if you look only at those blobs, you kind of zoom out and look at those blobs, then that gives you the tree of life, presumably. But now the question is, well, what does it really look like down at the level of this genealogical graph? Well, one thing you realize is species mating. There are two determinants of that. One is, well, are they the same species? The other is, do they ever even meet each other? You know, maybe one's living in Australia and one's living in Siberia, you know, and they're you know, some type of small rodent or something. The one in Siberia is not going to meet the one in Australia, at least not in that generation. And so there's sort of a spatial component to the whole thing as well. So there's both, in terms of our physics project, we talk about spatial separation of things corresponding to what happens in physical space. We also talk about branchial separation in terms of the branches of history of our universe. When we're thinking about that in terms of fundamental physics, it's all wound up with quantum mechanics. But what we have here in the case of evolution is kind of branchial separation is genealogical separation. Branchial separation is how, you know, you might have a 15th cousin, you know, 10 times removed or something. As you get a sufficiently distant cousin, that is branchial separation. That's branchial distance, is the number of cousin levels that you're going. And by the time, you know, I'm sure that, I don't know how many cousin levels you have to go before you reach non-human primates from us. I'm going to guess, let me see, there are 100 billion humans that have lived, and they've had, let's see, how many generations? Maybe, let's give us a couple of 100,000 years. We've got maybe 10,000 generations of humans. So I'm going to guess, yeah, I'm going to guess by the time you're at the 10,000th cousin, it's an ape, non-human primate. You know, and so you kind of have to go distantly on this branchial graph, branchial dimension of the graph. So I kind of have this suspicion that thinking about the dynamics of sort of branchial separation, spatial separation, and time, the actual, you know, what happens through time is a new organism is born, and then eventually a new organism dies. And actually, in most of nature, the new organism dies because it's eaten. And so then you have this kind of idea, there's an additional kind of network, which is the food web network, where an organism has, one organism eaten by another organism, which is eaten by another organism, on land. I think there's like 5 trophic levels or something typically of one organism eats another organism until you eventually reach, the apex predator, so to speak. And in the oceans, I think they're more like 20, 30 levels where, a little fish is, bigger fish. There's always a bigger fish that can eat the smaller fish, so to speak. But sort of the concept would be that one can imagine sort of the whole genealogical tree of everything that's ever lived and everything that's ever eaten anything, and that somehow the construction of that graph will have certain invariant properties, just as the construction of the graph that represents space-time has certain invariant properties that lead us to phenomena like gravity and so on. So it is my guess that that very large graph that corresponds to the possible configurations of all the organisms that ever lived will have certain universal properties that we can identify. One of those properties might be the very fact that species form. There may be other properties that we don't yet know that are sort of global facts about biological evolution that we can deduce, just like we can deduce global facts about space-time from knowing what, down at the level of sort of the atoms of space, there's this big network of connections between atoms of space. But if we kind of zoom out, there are big features like black holes, like gravity, like different aspects of space-time and so on that we can see. And maybe there are similar things that are true for kind of the genealogical or biological evolution structure that can exist in that case. And I kind of suspect something like that is true, and it needs to be worked out, and I haven't done it yet. And something maybe at our new Wolfram Institute, it may be one of the topics that we'll look at is biological evolution thought about in kind of this multi-computational way. Let's see. People asks, if mammals have a common ancestor, how did they get divided into carnivores and herbivores? That is a good question. Well, remember, there's something in the middle, like us. We're omnivores. We've got both the pointy teeth and the grinding teeth. We eat both vegetables, or some people choose to eat only one or the other, but most people eat both vegetables and both plants and animals. So there's sort of an in-between. And so my guess is that there were precursor organisms, mammals even, that had various tendencies towards omnivorousness, or like, well, I mostly eat meat, but gosh, if I see that particular kind of juicy fruit, my teeth will allow me to eat it. And I eat it and it works out well, kind of thing. And then there's gradual sort of evolution in that direction. So I kind of think it's not the case that, there's sort of kind of a gradualness that you can get from carnivores to herbivores, and the way you see that is literally in the shapes of teeth. The carnivores have pointy teeth for cutting meat. The herbivores have flattened teeth for grinding vegetable material. So that's my guess about how that works. Let's see. John here is asking, what do you think of the notion of chemical interspecies communication? Well, there's interspecies communication at some level. I mean, you can see the dog wagging its tail. You can see the expressions on the face of the dog. You can hear the barks of the dog. There is some level of interspecies communication that works that way. In terms of a sort of finer grained interspecies communication, where you're really transferring the great thoughts of one species' civilization to another, we don't know how that works, if it works. But certainly pheromones, molecules that are kind of secreted by one organism, intended as sort of a way to communicate with other organisms, those might have more detail in them. So, for example, a typical, they're typically molecules. I think they're often particular kinds of molecules that don't occur so often in other places, like an length 11 long chain of carbon atoms with hydrogens connected to them is a type of pheromone. and used by insects, I think, that particular one. And in a sense, it's like different, forms of perfume or something. You can have these different molecules that are sort of representing different kinds of things. So some animal, dog, well, I don't know, maybe skunk in a more extreme case, but these different kinds of animals will sort of mark out their territory by laying down these molecules, these pheromone molecules that sort of communicate to other organisms. Now, one thing that's true is that, that organisms have a certain molecular diversity. So, the smell of one organism is different from the smell of another organism. That's why you can have, a dog, you kind of have it, smell something from, that picked up the scent of a person, and the dog can then go track that person and so on. That happens because we all have slightly different, it is something determined by our genetics. We have, we're shedding different molecules that lead us to smell different, so to speak. And that's what, and there's great diversity in that, which is why you can expect, the dog to go find the right person and so on. And so that exists in all kinds of organisms. This idea that there's sort of diversity in the smells of different members of a species, and that is sort of something that can be used to kind of mark out territory and so on. I don't know the extent to which there is control of the molecules that are produced, whether there are examples of that, where you can kind of lay down for example, you could imagine, okay, the extreme, this is a good science fiction insect or something, is it writes its great works in pheromones on the ground. So in other words, it's it's going around, it's sort of hanging out, it's walking along, it walks along one line, then the next, always producing, secreting some different pheromone, some different molecule of some different scent. And as it's doing that, maybe it's drawing its great piece of artwork in smell space, so to speak. We don't know whether that happens. I certainly don't know of it, but I don't think we would know of it because we don't have good ways of detecting these things. I mean, we don't have good, artificial noses. We have good artificial eyes, namely cameras, which do the same kind of thing of detecting light that our eyes do. But when it comes to noses, things that are detecting all these different shapes of molecules that have different smells, we don't have good ways to do that yet. And it's still quite unclear how it works. I mean, probably in our noses, we have different receptors, different molecules there that have different sort of holes in them that fit different shaped molecules. So when you smell some particular kind of thing, you smell some particular kind of flower or something like that, there will be molecules there'll be molecules in the gas that come out of that flower, and those molecules will be fairly small molecules, probably if they're in gas phase, but they will then kind of lodge themselves into these molecules that are in your nose, and those molecules in your nose probably fit only molecules of a particular shape. And so what happens when you smell a particular smell is you've encoded in your brain, oh, there are sort of detectors in this region of my nose that are associated with the green smell and others that are associated with the purple smell. Now, obviously, smells don't naturally have colors, but you can imagine, just like in the eye, there are three kinds of cone cells that detect basically red, green, and blue. And in our brains, we've wired up the red cone cells to be the things that allow us to sense red in light. And so similarly, one imagines that we've wired up the nose cells, so to speak, that allow us to detect a particular kind of smell. We've wired those up to the place in our olfactory bulb in our brains where where we're kind of disentangling what smells like what. It's a complicated story. I mean, the tongue, for example, with taste buds, has its own complicated thing where there are these little sort of cavities where little particles of food get stuck there. There are different kinds of cavities that sense different kinds of sweet and sour and so on taste. It's not well understood yet how that all works. It's not well understood the extent to which it's sort of a chemical reaction that's happening in the taste bud versus the kind of the physical arrangement of the molecules that are just sort of fitting into crevices in the molecules in the taste bud and so on. But so, we don't really have a great way of doing what, dogs do pretty well. Of, a dog, if there was the great artwork put by the science fiction insect on the ground, the painting painted with pheromones, a dog might have a much easier time. It'll just go sniff, 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 and it'll walk down the lines of the painting and so on. Maybe it'll lose interest because it doesn't know what the heck it's smelling. But it would have a much better chance of having oh wow, that's a really cool painting, so to speak, than we would, because we just don't have our own sense of smell is not good enough to be able to distinguish things on that scale and so on. And we don't have automated ways yet of having sort of good artificial noses where we can detect those different kinds of things. For any particular kind of molecule, you have a CO2 detector, you have a carbon monoxide detector, you have a detector of some particular kinds of volatile chemicals in the air and so on. For some very particular kinds of molecules, we can make good detectors where you can get an electronic signal from the presence of that kind of molecule. And usually that's done with electrochemistry. Usually it's done by having some chemical reaction that has an electrical effect, sort of a very inverse version of a battery, where you're using electrochemistry to produce electricity. You're using chemistry to basically, well, I suppose it's the same thing, really. You're using some kind of chemical process to produce an electrical signal that allows you to detect the presence of that kind of molecule. But we don't know kind of a broad spectrum way to do that. We've had, you could imagine a camera which just has sort of one pixel. It's just, is there light or is there not light? But in fact, we can have these arrays of pixels, millions of them, and red, green, and blue detecting pixels, and even hyperspectral cameras where we detect different kinds of frequencies of light and so on. So there's kind of a whole, that's something where we can do it. For vision, we can do it in this very broad spectrum way. We don't yet have the technology to do that for smell, for olfaction. When we do, maybe we'll discover that, dogs have been writing big messages to us all along, or something like that. I don't know. It's an interesting case where sort of the things that are out there in the world could be a lot different than we know, because just our senses have detected only some aspects of the world. All right, I should probably wrap up very soon here. Oh boy, there's a question from M here. Can we think of some fungi species that could reach some kind of intelligence in the future like humans? Well, you see, the thing about stuff like that is, there are fungi that are these very, extended organisms that stretch over large regions and have just a lot of material in them. And maybe there are fungi that have some very complicated transport of material inside the fungus. Maybe there's electrical activity, maybe it's almost like brain activity that's happening in the fungus. And we could, at a scientific level, we could say we can measure this, we can see something's happening. But if we say, is that fungus thinking? Is that fungus making great works of fungus literature? We don't have a clue, because there isn't really an alignment between kind of our way of thinking and the fungus way of thinking. And even for the things I was describing about smell and so on, our way of thinking about things is very different. It's very visual. For example, we will, write out things in, on pieces of paper or whatever. It's very different from something where we do that for people to be able to see them. So it's very difficult to tell, whether there is a great civilization in the fungi and we just don't have any idea. And at some level, I think there is. It's just far away in terms of sort of the interpretation of what's going on from what we're used to as humans. All right, we should probably wrap up here. There's one here that maybe is fairly simple from Pippo. Are bubbles round because of gravity? No, it's not because of gravity. The bubbles are round because of surface tension. They, okay, first a math fact. If you are going to enclose the maximum area with the minimum perimeter, the way to do that is make a circle. As soon as you have corners, you're sort of wasting a fence. You're wasting a piece of fence by going out to that corner. If you want to, keep in the maximum area, the best way to do it is to have a circular fence. As soon as you push out a corner, you're using more fence, but not including much more area. So so another way to put it is, given a particular area, if you want to have the border of that area to be as small as possible, you're going to pull it in until it's a circle. Okay, so the same is true in 3 dimensions. The best way to enclose a volume is with a sphere. And so what happens in bubbles is that the soap is trying to enclose the maximum volume with a minimum surface area of soap. Why does that happen? It happens because of this phenomenon called surface tension. And how does surface tension work? What's essentially happening is that molecules in the surface are pulling on each other, and they don't have anything to pull. So if you extended beyond that surface, there will be much more force pulling the molecules into the surface than pulling the molecules out of the surface. And so that will make it be the case that you end up sort of always trying to pull the molecules together in the surface. And that leads to the surface tension, which leads to minimizing the area of the surface. And that's why soap bubbles are more or less round. Now, another thing that's round is something like a star or a planet or a drop of water that's falling. Actually, these are different. So, okay, the drop of water, it's a surface tension story. Same story as with, and the reason that happens is the molecules inside the drop of water are always pulling on their friendly molecules, say, come into the interior of the water. We're hanging together as a liquid. So anything at the surface, if there was a tower that developed on the surface, it wouldn't survive because all the molecules inside the water will be tugging on that tower to pull it into the interior of the water. And so it would end up getting smoothed out and become a round blob of water. Now, it also has to be said that under gravity, that gravity wins against surface tension. If you have a drop and that drop is and you put it on a surface, it'll kind of smoosh out because gravity is pulling on it and preventing it from being that spherical shape. Now, there's another phenomenon which is called wetting, W-H-E-T-T-I-N-G, which is the question of when you have a solid surface and a liquid drop, just how much does the solid surface cause the liquid drop to kind of spread into the solid surface? And it's quite common when you have some kind of, well, there are many materials which have this feature that they tend to keep drops from spreading into them. Some materials are very absorptive, some solid materials are very absorptive, and the drop will just quickly kind of decay and get spread out into the solid material. Others, you'll kind of keep the drop because the solid material will kind of say, we don't want pieces of liquid in the solid, so the drop is maintained in that form. But the reason that things like stars and planets and so on, the reason that they're spherical is because of gravity, because what's happening there is, in the end, it's the same effect, that it minimizes the surface area, but it happens for a somewhat different reason. It's not happening because the surface is sort of pulling things in. It's happening because the whole volume of the material is kind of acting on every part of the there's mass inside the thing, and that mass is having a gravitational effect that goes all the way out to the edge of the object, and that gravitational effect is making things as close as possible to the center, and the way to make a certain amount of material as close as possible to the center with a given amount of material is to make it a sphere, because you're kind of having layers of that sphere going out, and every time you add more stuff, it's like, well, we want to be a sphere, we don't want to be a tower that's stuck up. But the difference between gravitational forces is they're long-range forces, where the center of the star, the stuff at the center of the star is exerting a gravitational force on what's at the surface, whereas in surface tension, it's a local thing on the surface, that there are forces purely between things on the surface of the object that are causing it to have minimum area surface, as opposed to throughout the volume of the thing. All right, I think I should wrap up here. So thanks very much. I see lots of questions that we'll save up for another time. Well, thank you for joining our 100th Science and Technology Q&A for kids and others. I remind people there is a way of submitting questions for future episodes. I also remind people that we now have 100, as of today, 100 recorded episodes, where I've talked about just a huge range of different kinds of things. Thank you for asking me about different kinds of things all the time. And you'll find those things on the web as podcasts and so on. I do wonder, at some point, sort of, I will have said everything I know in one episode or another, maybe. And at least everything I know about these kinds of science and technology questions. But I think we still got a ways to go. So I think and I also learn new things every day, every week. I learn new stuff, and I have to say what I tend to do as I try and answer things here is I do I don't really have big flags for whether I learned something yesterday or 50 years ago, so I'm usually making a mixture of things that I've known for a long time and things that I've learned only quite recently. And also, I try to give some indication when things are things that are very recent facts that have only been discovered scientifically recently versus things that have been known for a long time. But there's a big mixture of those kinds of things. And the questions people ask here, there is a kind of a wonderful mixture of things where it sort of should be answerable by anybody with decent science education, and questions where it's like, gosh, well, actually, I can answer that because I recently talked to the world expert in that area, or because I've worked on that problem myself, and I kind of know what the absolute frontier of that is. It's kind of a thank you guys for giving me a wonderful mixture of those different levels of sort of scientific questioning. All right, well, I should wrap up here for now, and hope to see you another time, episode 101 and beyond. Bye for now.